Hello viewers, this is our protein synthesis model.
We'll be talking about DNA structure, transcription, RNA processing, and translation.
First, let's look at our DNA structure. As you can see, it's a double-stranded and a helix shape.
Here is our key. Our red gummy is adenine, orange is thymine, green is guanine, and yellow is cytosine.
For other parts of the DNA structure, the phosphate is represented by a purple bead and the deoxyribose sugar is the red vine.
Our model has complementary bases. Adenine bonds with thymine and vice versa, and guanine binds with cytosine and vice versa.
Adenine and thymine are bonded by 2 hydrogen bonds, which is represented by a purple toothpick. And guanine and cytosine are bonded with 3 hydrogen bonds, represented by a blue toothpick.
Our DNA model also has anti-parallel 5' and 3' ends. This is our 3' antisense strand, which we can identify because of the Tata box and the sugar end, which shows it is a 3'. The other side has a phosphate at the end, which represents the 5' end. And our antisense strand has the following sequence: TAT, AAAAGAT, ACGAATTT, ACGATAGGCACTCCC.
Now we will show the making of RNA. The process by which RNA is made from DNA is known as transcription.
Transcription must occur because DNA is too fragile and important to risk leaving the nucleus. The cytoplasm contains enzymes that can break DNA apart. So, the cells send RNA into cytoplasm as a copy of the genetic information, leaving the actual DNA in the nucleus.
In prokaryotes, transcription occurs only in the cytoplasm. In eukaryotes, this process happens in the nucleus, mitochondria, and the chloroplast. They both occur during G1 and G2 of interphase. The enzyme that is going to perform this process is RNA polymerase.
The anti-strand is the strand that is going to be transcribed.
There are 3 major steps for protein synthesis. The first step is regulation of gene expression. This is the process of turning genes on or off, meaning the genes are accessible for transcription or not. This occurs because it ensures that the appropriate genes are expressed at the proper times and in the appropriate cell types. Along with this, it can help an organism respond to its environment by only making proteins that are necessary.
Gene expression consists of multiple parts of DNA. A promoter, which is the sequence near a gene that RNA polymerase binds to. An enhancer, which is a sequence that turns on transcription when proteins bind to them. And a silencer, which is a sequence that turns on transcription when protein binds to them. It is also proteins that regulate gene expression. Transcription factors, also known as activators, which make the DNA accessible for transcription. And repressor proteins, which prevents transcription for the lactose gene. We know DNA is wrapped around a core of 8 histones. Methylation of DNA causes the nucleosomes to pack tightly together, which means transcription factors cannot bind to the DNA, and the genes are not expressed. Acylation of DNA results in the DNA to pack loosely. This allows transcription factors to bind to the DNA and genes are expressed.
The next major step is transcription. Transcription has 3 major steps itself. The first is initiation. In initiation, RNA polymerase attaches to the DNA at a sequence called the promoter to begin transcription. Our promoter is the sequence TATAAAA, otherwise known as our Tata box. A transcription factor or factors attach to the promoter sequence to tell RNA polymerase to start transcription. This gene sequence of DNA codes for a protein, which is what needs to be transcribed. The next major step of transcription is elongation. To know only one strand codes the RNA. This is the antisense strand, the non-coding strand in the sense strand. RNA polymerase moves along the DNA and breaks the H bonds between the strands.
It then builds RNA by adding complementary RNA nucleotides to the antisense strand. It is important to note that an RNA is single-stranded, and the thymine is replaced by uracil, which in our model is represented by a white gummy. Along with this, despite the sugars in RNA and DNA looking similar, DNA's sugar is deoxyribose, and RNA is ribose, which has an extra oxygen. RNA nucleotides are added by a 5' to 3' direction and is anti-parallel to the anti-strand. The phosphate ending in 5' and the ribose ending is in 3'. When RNA nucleotides arrive, they are triphosphates. Two phosphates break up and are used as energy to fuel transcription. The RNA strands break off and the DNA bonds again.
The final step of transcription is termination. At the end of the gene, there is a sequence of DNA called the terminator, where RNA polymerase will drop off the DNA. In our model, the terminator is a sequence CCC. After this, pre-RNA will move to the next major step of protein synthesis, RNA modification.
During RNA processing, the RNA is modified before leaving the nucleus because it needs to protect the mRNA from enzymes in the cytoplasm that attack the mRNA.
RNA goes through splicing during RNA processing. During RNA splicing, non-protein coding regions of the RNA strand are removed. This process must be done because it removes non-protein coding regions and allows alternate splicing. RNA processing is happening in the nucleus. This process occurs in the eukaryotic cells.
The RNA strand contains exons and introns. Exons are the real gene, and it's what's expressed. Introns are the junk, and they're between sequences. In our model, the intron is AAUGC.
The SNRNPs attach between the exons and the introns. Then they begin to close together and become known as a group known as spliceosomes. The spliceosomes do the splicing process in the cell. The introns are spliced out, and the RNA and exons are joined together. This connects all the exons, and the exons then exit the nucleus. In our model, our intron is the AAUGC. This is what occurs. The nerves, after they come together, remove the intron. This connects the other exons together.
A G cap is added to the 5' end and a poly A tail is added on the 3' end. These are only done to protect the ends. Finally, the exon RNA moves to translation.
Now we move on to translation. Translation is the process of building a polypeptide chain of amino acids, guided by a sequence of codons on the mRNA. Translation occurs in the cytoplasm in both eukaryotic and prokaryotic cells.
Translation consists of messenger RNA, mRNA for short, which carries the code from the DNA that will be translated into an amino acid sequence. Transfer RNA, tRNA for short, which transports amino acids to the correct position on the mRNA strands. Ribosomes, which provide the environment for tRNA attachment and amino acid linkage and amino acids from which polypeptides are constructed.
Let's take a closer look at the ribosomes. The ribosomal subunits are constructed from two types of molecules, protein and ribosomal RNA, or rRNA for short. There are two subunits that consist of grooves where the mRNA strand and polypeptide chain fit in. The subunits form a functional unit only when they attach to a RNA molecule.
Ribosomes have three binding sites, which mRNA attaches to. It has an A site where the tRNA brings in the next amino acid is positioned, a P site where the tRNA holding the polypeptide chain is positioned, and the E site where tRNA that has given up its amino acid exits.
Now on to translation. There are three steps to translation: initiation, elongation, and termination. In initiation, small ribosomal subunits attach to the mRNA. of the start codon, AUG. Then tRNA with a sequence complementary to the start codon known as the anticodon brings the first amino acid MET to the mRNA. In this case, the complementary sequence to the start codon is UAC. The amino acid attaches with tRNA by the molecule aminoacetyl tRNA. From here, the large ribosomal subunits bind and the ribosome is then built and ready for elongation. It is important to note that there are multiple ribosomes translating the same transcript on the mRNA. This is known as a polyribosome.
There are three parts of elongation: codon recognition, bond formation, and translocation. In codon recognition, the mRNA codon is in the A site. tRNA with the complementary anticodon hydrogen bonds to the codon in the A site. The tRNA then brings the appropriate amino acid to the ribosome. Secondly, in the bond formation, the large ribosomal subunit makes a covalent bond, known as the peptide bond, between the amino acid in the A site and the amino acid in the P site. The mRNA molecule is then read and new amino acids are added to the 5' to 3' direction. After this comes translocation. In translocation, the ribosomal moves towards the 3' end of the mRNA strand. Because of this, there is a shift in the tRNA binding site from A to P to E. tRNA then leaves the ribosomes from the E site. Finally, this process is repeated over and over and over.
And finally, the final step, termination. Amino acids are continuously added to the polypeptide chain until there is a stop codon on the RNA. The stops can be UAA, UGA, or UAG. In our model, our stop is UGA. A release factor binds to the stop codon and releases the amino acid chain. When completed, the ribosome dissociates into the two subunits, and the amino acid chain can now fold into its secondary, tertiary, and/or quaternary structure to become a functional protein. Parts can also degrade into their constituent nucleotides, and it can further degrade into uric acid and excreted as urine, or recycled to make more RNA.
And that concludes protein synthesis.